Great. So, yeah, thank you, first of all, to the organization of this webinar for inviting me to talk here. I will be describing in vitro models for evaluating foods and bioactive ingredients that we have in INEA. In particular, for the bioavailability, microbiome modulation, and other parameters. First of all, let me briefly introduce what is INEA for those of you that don't know. We are a technology center based in Spain, and our mission is to bring value to the companies. We are very company oriented, and our function is to translate basic research done in universities and public centers to industrial reality. Yeah, to translate this and leading the innovation and technology development. Our priority areas are focused on food and health, food quality and safety, industrial design and production, sustainability, environment, and also consumer studies. And for that, we apply a variety of technologies, of course, food technology, but also biotechnology and a full range of other complementary technologies, such as electronics, communication, chemical, energy and environment, packaging, or nanotechnology. We are a center, as I said before, very company-oriented. In fact, the majority of our funds is coming from companies. It's 72% of the direct contract with companies, and the 28% is coming from competitive public funds. We have a multidisciplinary team of more than 200 experts and more than 700 associated companies, and we work for more than 1,500 clients. Our sectors of activity are mainly food, but we also extend to packaging, energy, chemistry, pharma, and cosmetic. Regarding the theme of today, the purpose of the in vitro models that we use in INEA, why use in vitro models? The first thing is the necessity to demonstrate the biological or functional effect of an ingredient or functional food or cosmetic product. This can happen at several times, for example, during the product development to select the most active prototypes that we are developing, or during an administrative authorization process, for instance, to justify the claims in the labeling, or for commercial purposes, to fill a dossier to sell the product to a customer. A second reason is that the in vivo trials with animals are banned in many cases. For instance, the cosmetics in many countries, you cannot use animals anymore. And there is a social pressure to reduce animal testing also in food ingredients and products. Clinical trials with humans are, of course, the officially recognized methods, but they are very costly in both time and money, and they have also ethical concerns. And in vitro systems can be a quick and cost-effective alternative and can serve as this first validation screening. What we have in INEA are improved dynamic methods, because in vitro testing is usually not so representative, and we have worked to improve those in vitro methods to make them more representative of what is happening in the human body. And we use two sets of methods. First of all, the in vitro dynamic digesters to simulate the human digestion, both at gastric, intestinal, and colonic fermentation levels. And we use them to test food, feed, ingredients, nutricosmetics, or pharmaceutical active principles. And we combine that with cell-based models to mimic target organ. It can be the intestinal barrier, but also brain cells, bones, skin, or whatever, to study the bioactive effects of the foods, ingredients, active principles, or cosmetics. This is the first one. This is the first model that we built. This is patent protected, and this is what we call our in vitro dynamic digester. It simulates the gastrointestinal digestion. It means stomach and small intestine. And with this equipment, we can check the gastrointestinal resistance of a probiotic, for instance, or a molecule, an enzyme, or whatever. The glycemic index, the satiation effect through the viscosity, and the bioaccessibility. And combining this equipment with the cellular models, you can see this transwell system, we check the bioavailability. That means how much of the ingredient is able to pass through the intestinal barrier and reach the plasma and to reach the target organ eventually. We can simulate populational groups, that means children, adults or senior, by programming the equipment differently, and even monogastric animals. We have an advanced version of this digester, and this one, which is a digester in which we have improved the stomach compartment by doing it three-dimensional and very similar to a real stomach, and also simulating the peristaltic movements. And with that, we can simulate the solubilization rate, for instance, of pharmaceutical active principle. And we can also differentiate between the three different parts of the small intestine and see how much of the ingredient is absorbed in each region. We combine that with, again, these cell-based models to check the intestinal transport. And also, we are developing some organ on a chip to simulate the intestine in this kind of dynamic system as well. A third version of the model is the one that includes the colonic fermentation also. So it has an additional 3 compartments representing the 3 parts of the colon, and we inoculate that with a pool of feces, and we simulate how the microbiome is evolving when adding a supplement or a food or an ingredient to the diet. Yeah, and through the application of metagenomics and metabolomics, we can see how the microbiome is evolving, and also we can check if additional generation of metabolites in the colonic fermentation is happening. The second set of models are the cellular models to mimic the tissue or organ of interest. And here you can find some examples of models or effects that we have checked before. We designed the model and the study depending of the functionality that we want to see. And here you can find here some examples. For instance, for skincare, there are wound healing, anti-aging, whitening effect, antioxidant effect, and some others. And for food ingredients, we have checked a lot of anti-inflammatory effect, antioxidant, cholesterol metabolism, satiation effect, muscular metabolism, or whatever is interesting for the company asking for this kind of tests. This is an application example. Those are a combination of skin cell models and specific biomarkers that we have designed to test the skincare function of cosmetic ingredients. For example, combining immune cells with and checking changes in the different cytokines, we can see the anti-inflammatory effect. This is for active cosmetics. We can also analyze changes in elastin, collagen, hyaluronic acid, etc. in fibroblasts to see anti-wrinkle effect and some others. Yeah, this is another possibility to combine cell models with microbial models together to check the effect of prebiotics, postbiotics, or probiotics in either cosmetics or other formulations. For instance, we can simulate the skin microbiome, the oral microbiome, the vaginal flora, or the gut microbiome in combination with cell models. This is an example that we have published, a study of the evaluation of health benefits of einkorn-based breads. Those are with those ancient grains breads that are so trendy now. And in this study, we measured the bioaccessibility of the bioactives of interest of different breads. And after that, we checked the anti-inflammatory activity in a cellular model. As you can see in the graph, all of the breads have an increase significantly in the anti-inflammatory effect. And the conclusion in this study was that the higher carotenoid levels in the einkorn wheats are than the modern ones, and also the evidence of anti-inflammatory effect in culture cells. So this has to be checked with the in vivo testing, of course, but these kind of studies can serve to see if there is one prototype that is better than the rest. This is another example of the evaluation of health benefits of grape extracts, in particular of the polyphenols in the cholesterol homeostasis. And in this case, we studied first the bioaccessibility percentage of the bioactives of interest in the different phases of the digestion. You can see here the mastication, the intestinal digestion, and the final bioaccessibility of the different polyphenols, the gallic acid, lactic acid, epicatechin, and so on. We did just first the concentration of each polyphenol and also the antioxidant capacity measurements, and also measure the cholesterol metabolism in a cell model to check the real effect in a cellular model. And as a conclusion, the gastrointestinal digestion increased this polyphenol bioaccessibility together with the antioxidant capacity, and we checked that also observing change in biomarkers related with cholesterol metabolism. Yeah, and to finalize, I would like to see how we cooperate with companies. First, to say that this technology that I have shown before, the in vitro dynamic digestion, has been tested and validated with in vivo testing for some combinations of food and ingredients. We have active collaborations with food and cosmetic companies as well as research centers. We usually do joint research projects, but also we can work as CRO with research and development services, and also consultancy or legal services. Other related technologies that we have and did not present today due to the limited time is microencapsulation technology to increase the bioavailability of ingredients and compatibility with food matrices. Also the supercritical CO2 extraction to obtain high purity ingredients. We have even an industrial plant to do that. And the microbial biotechnology to obtain bioactive ingredients for microorganisms or through biocatalysis. Just a quick view of the legal aspects. So we have a dedicated legal department to help companies to bring products to the market with a specialization in Spain and the European Union. And they offer also consultancy and advice in labeling, authorization for commercialization of products, preparation of safety dossier, for instance, if it is a novel food, and preparation also of scientific evidence of health effects, for instance, to justify a health claim to put in the label. I will recommend to see the help desk of EFSA, which is the European Food Safety Authority, which is the organism that authorizes the new foods to be commercialized in Europe, and also the Spanish Registry of Sanitary aspects to be authorized to commercialize in Spain. And that will be from my side. I remain at your disposal for questions in the next section of the webinar. Thank you.